Good morning. Today I will explain how to use the Minitab to analyze factorial experiments or data that were obtained from factorial experimental design. Factorial experiments are widely used in both applied and social sciences. For instance, here we have an example of factorial experiment in which the weight loss was studied under two different diets and in both male and female. So we have the gender as one factor and the diet as another factor. We can have also more than two factors, but the analysis will be complicated. Let's go for another example and do the analysis. Here we have an example of factorial experiment. In this experiment, they studied the effect of different fertilizers on the growth and development of two plant species. The fertilizers in one experiment, the fertilizers were control F1, F2, and F3, and the species are two species each. So the treatments in the factorial experiments are the combinations of the levels of the two factors, which is 2 by 4, which is 8 treatment. Each treatment was replicated 6 times. Therefore, we have 48 observations or 48 experimental units in this experiment. Now we will go and see how to enter the data. I've already entered the data in the Minitab. Let's have the original data here. Okay. So now we have the first column is for the factor 1, which is the species A and B. 48 observation, 24 for A and 24 for B. Then we have the other factor in the other column, which is the fertilizer. We have 4 levels or 4 types of fertilizers, 3 and the control, and 6 replicate, and our response variable, which was the weight. Now, for instance, first we entered the A species in the 48 cells, in the 24 cells, sorry. Now, the first treatment was A with the control, which was replicated 6 times, and these are the observations from each replicate. So the first observation was from replicate 1, 21, the second 19.5, here. The third was 22.5, etc. Then we will move to the other column, which is the A with the F1, the treatment A with F1, which is treatment 2, and these are the observations that were obtained from the experiment. So this is the way we entered the data in the Minitab spreadsheet. Now we'll go for the analysis. Stat, ANOVA, general linear model, fit general linear model. Our response variable was the weight. The factors were species and fertilizer. We will check the model. Now, in the terms in the model are the two factors, but the interaction was not here. So we have to enter the interaction by mark the two factors, add the interaction. So now we have it in the model. Now, okay. All right. Here is our analysis. Now we have factor information, two factors, species and fertilizer, two with the levels of each factor. Then we have the ANOVA. Source of variation is species, fertilizer, species by fertilizer, or the interaction. This experiment is designed in completely randomized design, so we don't have block in the model. Also, it's clear that the main factors and the interaction was significant. Therefore, we go forward to the mean separation, general linear model, comparisons. We use the Fisher test, and we mark all the factors and the interaction because they were significant. All right, so species, fertilizer, and species by fertilizer. Now, okay. Then we have the output here. Mean separation for the species, mean effect. The mean separation for the fertilizers, F3 was the best. And the most important thing is the mean separation for the interaction. So we have the treatment B or species B with fertilizer 3 was the highest in weight. So it got an A letter, and so on. So we have these are the 8 combinations, the 8 treatments separated by the LSD. Now we'll go back to the to our data. Okay. Now suppose that our experiment was arranged, our factorial treatment design were arranged in randomized block design. So instead, for instance, for the replicate, we will have the block as a factor. So the analysis will not differ. Go for the ANOVA, generating a model. So we will enter the block as a factor. Sorry, here is the weight, but in the factor we have to enter the block because it will appear in the model. We'll go to the model now. We have already the interaction, the block, but no, these are the what will appear in the ANOVA: species, fertilizer, interaction, and the block. Now we will, okay. Here are the analysis. Again, we have 3 factors now, but the interaction is only measured for the species and fertilizer, species, fertilizer, block, species by fertilizer. So the design now is block design. So we have the block in the ANOVA, but the block was not significant. The same thing will happen for the mean separation, same way that we did with when the experiment was in completely randomized design. And the results will not differ. Okay. All right. Statistic, ANOVA, generating model comparison, and we will compare only significant effect. All right. Block was not significant. Okay. The same output that we obtained when the experiment was in completely randomized design. Thank you.